Okay, so before we get into the details, I just want to remind you, so this is something I covered in detail a bit in the chapter 3, but in order to get started, you really have to understand what the parental gametes. So in order to understand linkage, you have to know the order of the alleles in the parents that gave you, these alleles. So this is two different scenarios, and so in that activity, we talked about there's two different ways to do a dihybrid cross. So one of these was a genotype like this, and one of these was a genotype like this. And in both cases, you end up with a heterozygote that has one allele of both. Okay, so this F1 in both scenarios has the same genotype, big A, big B, little b. And so you might not think it matters what the parents are, but it matters a great deal now that we're looking at linkage. So in these two different scenarios, what's important to remember is that this individual, so let's say this is the father, provides a gamete with this particular genotype, and then the mother provides an egg, an oocyte, that has this particular genotype. But in this opposite scenario, the genotype of the sperm cell is different, and the same thing is true with the oocyte as well. So these have different gametes, and that actually makes a difference. So this is what we refer to as parental gametes. So we're going to talk about this throughout the chapter, this word parental gametes, and this is the order of alleles that occurred from the parents. And so you can define this, you can figure this out based upon knowing what the genotype of the gametes that produced an individual are. And so these are kind of two different situations that produce opposite gametes. So again, this individual has the same genotype, big A, big B, little b, but this actually motivates a different way to write out the genotype of an individual. So we want to indicate what the parental gametes are, and so another way to write this genotype of this individual is to show the gametes over each other. And so we write this as big A, big B over little a, little b. And again, this just comes from this right here. This makes this right here, and then this makes this right here. And so the same thing we can do in the second scenario, and so you can see that the genotype is actually written differently, big A, little b over little a, big B. And we'll see why this is important in a little bit. So genes are on the same chromosome, and in this case, again, we have an individual that has the following genotype, big A, big B over little a, little b. And so if we draw a picture of what those chromosomes look like, it looks something like this. And so what I'm showing you is something from prophase 1, where the tetrad has form. And now it is possible to actually produce hybrids between these two parental combinations. And so if we didn't actually have crossover, if we didn't form the tetrad, it would be impossible to create new combinations of these gametes in offspring. And so this is one of the reasons that this has evolved, is because it's actually beneficial to have unique combinations of alleles in offspring, including the situation where the genes fall on the same chromosome. So, if we didn't have crossover, this would be one of the gametes that could be produced. This would be one of the gametes that could be produced. And so that is kind of what we're showing here and here. But now we have a crossover event, and so what is created is you just follow this going through here kind of to the other side. And so this right here would create this gamete right here. So we're kind of color coding where exactly that crossover event has occurred. And then we've also done the same here. So this right here could go this way. And so that would create this gamete right here. And so what we end up having is the original parental combinations, but then we also have these new crossover gametes that produce these new combinations. So again, if we start out in the situation where it's like a dihybrid cross, we can produce all 4 of these gamete combinations. And those are produced at an equal ratio. So this is produced at a quarter of the time, this one is produced a quarter of a time, etc. And so the parental gametes, again, would be these 2 right here, because that's the original combination. And then the recombinant gametes would be these 2 right here. And so in this situation right here, when they're on different gene, you have 50% recombinant and 50% parental gametes. Now let's consider a situation where there is no crossover. So you're unable to do crossover. So in this situation, again, the original combination is big A, big B, so they're on the same chromosome. But because crossover can occur, there's only 4 types of gametes that are produced during meiosis. So you split during anaphase 1, you split these out, and so then you split them again, and then what you're going to create is, so if this is not clear, just kind of go through the process of meiosis just to make sure you understand it. But what you're going to do is you're going to create 2 of the big A, big B, and then 2 of the little a, little b. And so in this case, you produce 100% parental gametes and 0% recombinant gametes. And this is kind of just like a thought experiment right now because we know crossover exists. But if there is linkage and there is crossover, then we can produce both. And so the frequency of this depends on how far apart the 2 genes, because basically what you need to know is what is the probability that a crossover occurs between them. So potentially you could have a crossover occur outside, either to the left of A or to the left of B. And so in that case, this is where it becomes a percentage where the parental are the most common, so the parental gametes are always the most common, between 50 and 100%. And then the recombinant gametes are produced between 0 and 50% of the time. And so this just kind of shows you, 2 situations where you have genes that are close together and then genes that are further apart. And so the frequency of the recombinant gametes is much lower in this situation because you have to have the crossover occur in this very small region of base pair DNA. So this motivates a definition of map distance, and we'll see how to calculate map distance in a second. But if we measured this, so if we took this cross and we cross these individuals together, so this actually falls on the X chromosome, but we have a female that is heterozygote, and we cross it to a yellow-white. This individual will produce 4 different types of gametes. So this individual can produce 1 of the parental gametes right here, 1 of the parental gametes right here, and then it can produce 2 of different types of recombinant gametes right here and right here. So by crossing it to this male individual, we can measure the genotype of the gamete produced, because in one situation you would be providing the Y chromosome, or actually in Drosophila flies, it's an XO system, so that you just would not have a chromosome here, and so you would see wild type based upon the genotype of the gamete. And so in this situation you would have white, white, and then yellow. And so this allows you to measure the frequency of the recombinant types, and so we can measure that at 0.5%. So the majority of parental gametes the majority of gametes are parental gametes, and that occurs about 99.5% of the time. And then this is the females. In the female situation, you can also see what it is, because you know that you always have the recessive alleles provided by the male gamete. So in this situation right here where they're further apart, you're much more likely to end up with a recombinant in between the two genes. And so the recombinant type is 34.5%. And so this motivated a definition to define the genetic distance, how far apart two genes are, based upon this rate of recombination. So this is called a map unit, sometimes abbreviated as MU, and it's also called centimorgan. So centimorgans is another unit of measure for map distance. And so it's defined as 1% recombination between two genes on a chromosome. So how often you see recombinant gametes. So these are relative distances, not exact distances. And then in genes that are 50 map units apart, crossing over can be expected between 100% of the tetrads. And so what that means is that it acts the same way as a dihybrid cross, where you have 25% of each of the possible gametes. All right, so how do you measure map distance? So in the first case, the genes are on different chromosomes. So you always start off with a heterozygote individual, and you look at how what the frequency of gametes that are produced. So in this case, the parental gametes are produced 50% of the time, and the recombinant gametes are produced 50% of the time. So you would measure, out of 1,000 gametes, and then you would divide the number of individuals that were recombinant, and then that would give you your measure of percent of recombinant gametes, and then that would be approximately 50 centimorgan. All right, now we have two genes that are linked. So this is the parental combination, and then we have some sort of way to measure the genotype of all of the gametes that are produced by this F1 individual. And so we add up this. So 25% of the time you form recombinant gametes, multiply that by 100, and so the map distance in this case is 25 centimorgans. So how do you actually estimate this? So how do you actually estimate the genotype of a gamete? A sperm and an egg don't have a phenotype. You can't look at the sperm cell and say, okay, this is a yellow eye fly. This is a ebony colored fly, because you don't see the phenotype until you actually have an animal. And so what we do is we use a test cross to measure this. Again, test cross was covered in chapter 3. You should be familiar with a test cross. But the idea is that you cross with an individual that is homozygous recessive. So we have this individual right here, big A, big B, little b, and we cross it to the recessive individual right here. So this individual right here can produce 4 different types of gametes. So it can produce big A, big A, little b, little a, big B, and little a, little b. This individual can only produce 1 type of gamete, little a, little b. And so I'm just showing you basically what the test cross genotype is, the genotype of the offspring, if this was fertilized with this, if this genotype was fertilized with this. And so these are the 4 genotype combinations that you can have. So in this situation, so let's say this is an individual that is wings and white, and this individual would be no wings and ebony. So if we looked at the phenotype of this individual, this would be wings white. This would be this would be wings ebony. This would be no wings white, and this would be no wings ebony. And so we can use this phenotype of these offsprings, we can measure the phenotype of the offspring in order to determine what the parental gamete was from parent 1. And so let's say we did this cross and we isolated 1,000 total flies and we measured their phenotype. So we would group all of the winged white flies together and we would say, okay, this is how many times this big A, big B was produced, 400 times. And then we would do on the same, and then we would fill in this for all 4 of the different possible genotypes. So if we had 100 flies that were winged and ebony, then we would say there was 100 big A, little b gametes. And then 100 no-wing white flies, 100, and then 400 no-wing ebony flies, 400. So this data right here, one, it allows us to determine what the parental gamete combination was. So the parental gametes are always the most common. So that's a rule that you should remember, is that parental gametes are always the most common. And so this genotype of this fly can be rewritten as big A, big B over little a, little b, just to indicate the parental combination. And now we add up how many recombinant gametes there are. There's 200 divided by 1,000, and so the map distance is 20 centimorgan. So always connect the map distance with the recombinant gametes that are produced, the ratio of recombinant gametes that are produced. So this can occur, so this is an example of a single crossover, and so the recombinant gametes that are produced here, you just kind of follow this right here. So these occur between non-sister chromatids. So this and this are non-sister chromatids. You can also have double crossover events. So in this case, you have 2 crossovers, where a crossover occurs here and here. And so the way you would figure out what sort of gamete is produced is that you would just simply follow again. You cross over, and then you would cross back over again. And so you end up having this orange, blue, orange pattern. And then depending on the gene locations, you can again flip the allele combination. So big A, little b, big C. So linkage in action, how does it affect ratios? So big A and big B are 20 centimorgans apart. What percent of the offsprings do you expect to have the recessive trait for both genes in an intercross of big A, big B, little a, little b? So how do you do this question right here? So this is kind of a way to modify the Punnett square in order to account for the fact that you no longer have a quarter, 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 quarter ratios. So we know the parental gametes are big A, big B, and little a, little b. And now based upon this map distance, we know that the recombinant gametes are produced at a 20% rate. And so now I've written, again, so the way you do a Punnett square is you write out the 4 possible genotypes that can be produced, the gametes that can be produced by this individual. And so now we're also adding the frequency of our expected frequency of how often they would be produced. So this gamete would be produced 40% of the time, 10%, 10%, and 40%. And because it's an intercross, this side of the square would be the same because it's a cross between 2 individuals that are heterozygous. And so again, we can do this. On this side, so 40% of the time this gamete is produced, 10%, 10%, 40%. And so now to fill in the square, we simply multiply these numbers together. So 0.16 equals big A, big A, big B, big B. 0.1 times 0.4, so 0.04 here. And so if we add up all of these numbers, they'll still add up to 1. And then, if you wanted to figure out the frequencies of a genotype, you would just simply have to group them together that share the same genotype. So the question that we asked, what would have the recessive trait for both genes? And so we're interested in this part of the square right here. And so the answer is 16% of the time. So this is how we now use this information about map distance in order to figure out how the ratios actually change. And so again, I use this information to figure out the frequencies of gametes produced. So that's always what you want to do with map distance, is go to the gametes that are produced. All right, so that's the end of part 1, and next we'll move into 3-point mapping.